That's great. Okay.
Perfect. So what I was going to talk to you about today is some, general news stories that we have working in epigenetics. I just moved to London 2 months ago from MSK. So we are restarting the laboratories here in London. It's very nice. People did move with me, and they're also coming a few people from Copenhagen, where I've had a position despite being at MSK the last 3 years. So the work in my lab have, mainly two different lines of research we're doing. One is very mechanistically based epigenetic projects, trying to understand what I think most of us try to do, the understanding of epigenetic machinery and its role in transcription control and cell fate. And the other is more regular, related to cancer, where we try to understand how the deregulated epigenetic processes can initiate contribute to cancer. And we also have some discovery type research here where we try to identify potential novel targeted therapies for cancer. So today I would like to tell you a couple of stories from my lab. One is not published, and the other one is in the works, and the other one is we published recently. One of them is on the functional roles of H4K27 methylation and acetylation in mammalian cells, and the other one is on the role of basically switching the coin, if you want, of the Histone acetyltransferase Cat8, which is also known as MUF or MIST1, and is containing complexes MSL or NSL complexes in transcription and cellular homeostasis. So it's a very interesting complex. So let me start with the Polycomb group proteins. And for those of you who are up like 4 o'clock this morning, if you're in California, you probably heard some about the Polycomb group proteins from Luciano Di Croce. And later we will also hear some from Luis later during this session. So the Polycomb group proteins, of course, most of you know, are really interesting proteins. They were originally identified in Drosophila as transcription repressors of developmental regulators involved in body segmentation. And you can see that on the pictures here. Here's the homoerotic genes expressed, and you see a deregulation of homoerotic genes when you knock out the Polycomb group proteins. It's also known that it's evolutionarily conserved in fungi, plants, animals, and you get very strong phenotypes by knocking them out, which is shown here as well. And one of the reasons why we started working on them, in fact, was that they were downstream regulated, or being regulated by the E2F transcription factors, if you like, came from cell cycle control. But it's also been shown that BMI1 was associated with cancer. One of the first to demonstrate that was Martin van Nieuwhausen when he was in Anton Bern's laboratory, where he showed that BMI1 collaborated with CMYC in inducing B-cell lymphomas. But in recent years, there's been more focus on EZH2, which is part of the Polycomb repressive complex 2, because it's been found mutated in tumors, and there's been developed specific inhibitors to EZH2, which are now actually used in the clinic. So the Polycomb group proteins have been studied by many over the last years, and I try to summarize what they do and what we've learned about that. So one of the things that we learned early on is that they are important for normal cellular proliferation and homeostasis. And what they do is to regulate genes in a way that they are plastic during development and differentiation. There are many genes which are regulated by the Polycomb group proteins. In our estimate is 3,000 to 4,000 Polycomb target genes in each individual cells, and these are the protein target genes and not the non-coding target genes. And that means that there are many of those genes regulated by the Polycomb group proteins in each individual cell. And of course, as we know, these are enrichment protocols, so the exact number we don't know. There are two major complexes of the Polycomb group proteins. One is PRC2 complex, the other one is PRC1. PRC2 and PRC1, they are enriched in CPD islands of silence genes, and they co-localize with their modification, if you want. PRC2 leads to catalyzes of H4K27, PRC1 at the mononucleosylation of H2A and lysine 119. Binding of PRC2, we and others have shown actually occurs in response to transcriptional silencing, but they are already set up, if you want, so they do regulate whether a gene is actually being activated or not. The question is their relationship to epigenetics. There's been models, including models from my own lab, which has suggested models for how you can actually propagate H4K27 methylation in the absence of DNA binding. But I think the jury is still out there, because we have also shown that H4K27 methylation patterns can be faithfully we established in the absence of prior H4K27 methylation. Clearly, they have a role. We know they have a role, but we're still discussing how epigenetic that role is. And we're also discussing the mechanism regulating the association of Polycomb group proteins with chromatin because it's extremely complex. And this is just a small summary of that, which is shown over here. PRC2 creates this H4K27 trimethylation mark, which is actually a mark also being recognized by the chromodomain of the CBX proteins in the canonical PRC1. However, we also know that there are variant PRC1 complexes which can actually directly bind to the CBD islands, creating an H2A ubutylation platform, perhaps which is involved in the regulation of binding of the PRC2 complex. So it seems to be a strong regulation here between the PRC1 and PRC2, which actually bind to these CBD islands, also depending on the modification, the post-translation modification of the nucleosome. And then, of course, I haven't talked about RNA. RNA is also involved in it. Also complex. In some cases, it's been seen to attract the PRC2 complex to certain sites, and other situations, it actually leading to dissociation. So if you focus in on PRC2 and H3K27 methylation, it's a little more less complex shown here. This is the core members of the Polycomb group complex, SUSI12EED and EZH1 or EZH2. It's the only known H3K27 methyltransferase complex in mammalian cells. In plants, we have other complexes, but not in mammalian cells. As I said, they're essential for normal development. They maintain transcriptional repression by actually giving rise to H3K27 trimethylation. We know that there are other modifications modified by this complex, monomethylation, dimethylation, and trimethylation, and these are extremely abundant modifications in the cell, basically saying to us that there's a scanning of the DNA and of the chromatin in each cell cycle, which leads to actually these modifications, and therefore a specific transcription factor is most likely not involved in the mono and dimethylation, and the trimethylation, we think that is actually they are retained there rather than as a recruitment mechanism. As I mentioned, in cancer, PRC2 is hyperactivated, and we see somatic mutations in EZH2, in lymphomas, melanomas. We see these dependencies, very relevant from the previous talk. Mutations in the SWI/SNF complex seems to be extremely dependent on EZH2. We see gene amplification in breast, ovarian, and prostate. And we, and also recently others, have shown that diffuse intrinsic pontogliomas, which express this variant of H3, giving rise to lysine 27 methionine, are extremely sensitive to EZH2 inhibitors as well. So the idea is basically that it leads to hyperrepression of PRC2 target genes with these increased sensitivities. However, we also know that there might be novel targets here which are actually repressed by the PRC2 complex, and therefore leads to their sensitivity to EZH2 inhibition. There's been an EZH2 inhibitor which has been approved for patients with relapsed or refractory lymphomas and for patients age 16 or older with metastatic or locally advanced epithelioid sarcomas, and these are the ones which have mutation in SWI/SNF. So one of the questions we've asked is whether we know the functions of the histone modifications. In this case, you know, are they really important? And I will tell you why we think this is an important question. How important are actually the methylation of H3K27 for the effect that we see of Polycomb group proteins here, PRC2, have on gene expression and on cellular processes? And so the work I'll present you now is done by two former postdocs in my lab, Faisal Mohammed and Dita Sankar, who left my lab in the last couple of years. And it's still a story that we're working a bit on in the laboratory to try to get it published. So why would we actually start looking at that? And we know this very much from kinases, that kinases have catalytic function, non-catalytic functions, plus they have extremely a lot of different substrates. And this has also been described for the histone modifying enzymes, which actually give modifications of methylation of lysines and also arginine. And here's just some reviews discussing that lysine methylation regulators, moonlighting outside the epigenome, the importance of non-histone methylation in cancer therapy, beyond histones, the expanding roles of protein lysine methylation. And this is actually a paper from our own lab, which shows that PMT5 methylome profiling uncovers a direct link to splicing regulation in acute myeloid leukemia, where PMT5 actually think that most of the most prominent modifications are actually occurring outside the histones. So the big question is, how important are these non-histone protein targets for biological effects of the lysine methyltransferase? In other words, are the histones the key targets of lysine methyltransferases. For PRC2 or EZH2, we know several non-histone proteins that are methylated by PRC2, for example, GATA4, Longan A, our alpha, and GAR2. And the question we asked ourselves was H3K27 actually the key substrate for PRC2, explaining its function for homeostasis and gene regulation. So how could you actually study the importance of H3K27 methylation in cells? Well, you can insert mutations in H3K27. This is feasible in Drosophila, very difficult in mammalian cells. Drosophila has a single cluster of canonical H3, which can be deleted or rescued by a single back. In mouse, we have 12 genes for H3.1 and 3.2, 2 genes for H3.3. That means that if you have to mutate them, you have to mutate 28 alleles. And for human, you have one more gene for H3.1, therefore you have 30 alleles you have to mutate. This would be H3.2. So the caveat here is, as well, is that when you do that, you also have an effect not only on H3K27 methylation, but also on acetylation, and if there are other unknown modifications of lysine 27. There has been some published data on that. Drosophila studies have shown that substitution of K27 on H3 to H3K27R or H3K27A in canonical, that is, the replication-dependent H3 leads to a Polycomb phenotype and the derepression of some Polycomb target genes. And these are studies from Jörg Müller's laboratory and Dan McKay when he was working with Bob de Runyo and also with what's his name, Greg Matera when he was in UNC. So in Drosophila, it has also been shown that substitution of replication-independent H3.3K27 with H3.3K27R was also found to lead to increased expression of Polycomb target genes. This is very interesting. You get very similar phenotypes. Whereas in mammalian cells, it's not been done very much. And the only study which I know of is from Bing Xu, was mutated H3.3K27R in mouse embryonic stem cells. And he shows in that study, or they show in that study, that is not required for enhanced activity. But we also know there's a lot of K27 acetylation left when you mutate H3.3. So what Faisan started to do in my lab was actually develop a strategy to generate mutations in H3.1 and H3.2 lysine 27 in mouse embryonic stem cells. And what he used was an adenine base editor, which was published by Liu's laboratory at MIT. And so basically, one of the things he did was to use this system, but at the same time, what he did was actually send up a single cell sorting strategy and then a qPCR strategy to identify clones with the most mutations. And he could do that because you could use the same guide RNA sequence to basically the 12 alleles here, which are shown here for H3.1 and 3.2. And by doing this mutation analysis and identifying clones, what he found were actually clones which had mutated all 24 alleles. And when he analyzed those further, which is shown here, these are known as canonical K27R, and there are 2 different clones. What you see here is a strong reduction in monomethylation, dimethylation, and trimethylation. We know there's some methylation left, but this is the Western blot or the sensitivity of Western blots. We also see some K27 acetylation using 2 different antibodies. However, we did not see major changes in other modifications on H3 or on any of the other histones we analyzed. So what Faisan also wanted to do was to make a pan-mutant. And what he did there was a little other approach because we didn't manage to actually mutate the endogenous H3.3 in these cells. So what he did instead was put in a balancer expressing exogenous H4.3 wild type or mutant K27R, where he knocked out H3.3 at the same time. And this is shown here. They do indeed express there, and what you see is trimethylation of those as well as the wild type allele. When you now analyze those to look for monodiene trimethylation, you see the loss, and you still see acetylation here, but you don't see the K27 acetylation when you basically analyze the pan-mutant. Again, when you look at what we were trying to compare to, what's the phenotype in these cells compared to when you knock out the whole PRC2 complex, we compared the methylation patterns and the modifications in SUSI12 knockout cells compared to when we actually had these knockouts. And what we know from previous studies from our lab is that you basically knock out K27 in those cells, but you see a huge increase in K27 acetylation, basically suggesting that one of the phenotypes we are seeing during development could be the gain of K27 acetylation. And in fact, it has been suggested what K27 trimethylation does is primarily preventing to have K27 acetylation. So we looked into what happens with these cells, embryonic stems or ES cells, when we differentiate them into embryoid bodies. We know from knockout studies from my lab and other laboratories is that when you mutate the core components of PRC2, you don't pass gastrulation, which can be mimicked by making embryoid bodies in vitro, where you basically form the free germ layers. So this is shown here for wild type, expressing Cas9 alone. You get these beautiful embryoid bodies. When you have SUSI12 knockout, you basically see that you have small bodies formed, which cannot really make it as well. When we look at the canonical one, you see bodies which are perhaps in between, they still form okay. And then when you see the pan, they are again more similar to what you see with the SUSI12 knockout. So of course, this is kind of a phenotypic assay, which may not tell us very much about what happens in individual cells. So what we also did was to look at gene expression pattern. This is a very busy slide, so the only thing I want to say is that what we see in the pan mutant and also in part in the canonical mutant is that we see failure to downregulate proliferating and stem cell markers, which we observed before by knocking out PRC2. And we also see failure to induce efficient expression of genes involved in formation of the three different embryoid germ layers. And here it might be actually, since we're not really executing pluripotents as well, this could be one of the reasons why we don't see basically the expression of these, which are specific for the embryoid germ layers. So the question is, this failure to induce differentiation due to changes in H4K27 methylation, acetylation, or both? And we try to address that by several different avenues of research. So one of the first one, which was relatively easy, is to see what happens to gene expression in mouse embryonic stem cells when you knock out SUSI12 or you actually exchange lysine 27 for arginine, either in canonical or the pan mutant, which is shown here. And what you see here is you see derepression of genes. And of course, you see also genes as secondary effects, they also get repressed. So there's no problem if you want to derepress or activate transcription in proliferating mouse embryonic stem cells. And so you don't have acetylation, particularly not in the pan mutants. So it's really what this suggested to us is that you don't need acetylation actually to have the transcription activation or the repression of the genes regulated by the Polycomb group proteins. We compared the profiles of the different cells, which are shown here, and they're very, very similar between the canonical, the pan, and the SUSI12 knockout. So it seems like that this overlap is also not only seen in the phenotype, but also when we look at transcriptional patterns. You could then argue that this is a place where we actually see derepression is not clear activation. So what we did as well was try to use an assay where we could look at specific activation of genes which were not expressed at all in the embryonic stem cells. And what we did there was actually differentiate epiblast ES cells to epiblast-like cells by actually growing them in activin and FDF for 48 hours. One thing is important here is that when you knock out PRC2, it does not seem to have major effects on the differentiation of embryonic stem cells to epiblast-like cells, whereas it does have an effect when you differentiate between embryo bodies. And when we did the same experiment with the pan mutant, we didn't actually see major effects either. And we didn't see any major effects actually on induction of gene expression either in these situations, where we see that basically we see that they are repressed, but we also see that there are specific epi-like stem cell markers which are going up during differentiation in these cells, no problem whatsoever. And if we looked at a genome-wide level, we basically saw a highly significant overlap of gene expression profiles of cells lacking SUSI12 and expressing H3 or expressing H4K27R. And basically what it looks like is K27 acetylation is not required for the induction of gene expression, suggesting that the major phenotype we're looking at is due to H4K27 acetylation. So I just like to summarize this part of my talk by saying that what we've shown is that pan mutant of H4K27R replicates the Polycomb knockout phenotype and transcription effects in the formation of embryoid bodies, and which is an in vitro surrogate for early development. The increased H3K27 acetylation observed in the Polycomb mutant cells is not required to elicit the Polycomb knockout phenotype. In other words, we think that H3K27 methylation, and particularly trimethylation, appears to be an important modification. And finally, I think, which is potentially not surprising, but H3K27 acetylation is not required for ongoing activated transcription in self-renewing ES cells and during differentiation. So H3K27 is the substrate to study for PRC2 methylation, whereas H3K27 acetylation may only be a biomarker for P300 CPP and enhancer activity. And of course, we know that P300 and CPP have many additional modifications, and it could be that H3K27 acetylation is one of the ways that P300 CPP actually contributes to transcription activation. Good. So then I'll go to my next story, and this is about histone acetylation. So now we go from gene repression to what happens when we activate genes. And this is major work from Alex Ratsithoyskaya, who's in my lab until Monday, where she starts her own lab actually here at the ICR. So this is very interesting from many points of view, because it goes deep down to what we're all been talking about, and what people prior in this session and also session talking about is actually looking at what is regulating the openness and the closeness of chromatin. So histone acetylation is a hallmark, as we know, of open chromatin. It neutralizes the positive charge of lysine residues weakening histone DNA interactions. It prevents adjacent nucleosome interactions, and is also being shown to recruit bromodomain containing proteins. So what we know as well, and this has of course been one of the big discussions about the histone code proposal, is that there seems to be a cumulative and redundant effect on chromatin of the acetylation. So the number seems to be an important one rather than the position which defines the response. And this is particular studies in yeast which have shown that, and not so much in mammalian cells. So what I want to talk about was H4 lysine 16 acetylation. There have been several studies which have shown that it affects nucleosome interactions in vitro, and it's also been shown that it's actually required for Drosophila dosage compensation in Drosophila, of course. And this is actually a very nice study from two different laboratories. Robin is actually my lab now, coming from Bob de Brunio's laboratory. And what she did here, and also the other group here, was actually substitute H4K lysine with an arginine, so they only express the lysine, so the arginine mutants in Drosophila. What is very interesting here is that when you look at that, H4K16 arginine actually does not lead to any males which are born here from the Drosophila, but it does lead to females. And of course, we know that H4K16 acetylation is involved in dosage compensation in Drosophila, and is particularly therefore involved in the transcriptional upregulation of the X chromosome in the male germline, and therefore it is required for there. But it does not seem to be important for the female, and therefore not for gene activation later, or perhaps chromatin structures later in Drosophila development in the female. Anyway, so we asked the question in mammalian cells: does H4K16 acetylation regulate chromatin accessibility in vivo, which is shown in vitro in these two assays shown here at the top of the slide? And what is the role of H4K16 in mammalian cells? So what is actually regulating H4K16 acetylation? This is a protein, or a histone acetyltransferase called CAT8. And CAT8 is actually incorporated in two distinct conserved complexes. One is called MSL, and the other one is called NSL. And this stands for male-specific lethal complex and non-specific lethal complex. And this is data primarily in Drosophila, which leads to this characterization. So we know that in Drosophila, MSL is involved in dosage compensation. And in Drosophila, we also know the other complex is involved in activation of housekeeping genes. So what's the functions in mammals? Well, CAT8 loss, if you want, induces apoptosis, autophagy, and a deficient DNA damage response. And cancer cells, we know, they have frequently exhibit low levels of H4K16 acetylation. So one of the questions we ask ourselves, since we have two complexes here which have the same catalytic subunit, how can these two complexes carry out different functions if they both lead to H4K16 acetylation? And of course, this could be specific recruitment, but it could also be that they have specific and different catalytic functions in these two different complexes. So we try to understand whether this complex is actually essential for proliferation in mammalian cells. And so this is the knockout using CRISPR knockout guidanase of the subunits of the NSL complex. They're called cancel 1, 2, and 3. CRS1 is also essential, whereas the PHF20 subunits are not. These are not specific for this complex, but they are also lethal. Whereas what you can see here is that the MSL complex, the cells that grow happily without them, whereas Cat8 is actually essential for the proliferation of these cells. So we try to map basically where the NSL complex is enriched in mammalian cells. We found that cancel 3, which is one of the subunits of NSL, is actually mapping on transcription start site. And what we also found that there's a very strong overlap, actually identical overlap with 70% of the peaks with H3K4 trimethylation, and also the rest of them were actually very close to transcription start site. We knocked out NSL, as I said, in human cells. We knew that they were actually essential for it. So instead, we did a knockdown to look at how does it actually regulate transcription in human cells. So for doing that, we use the CRISPR interference system, which is shown here. We actually it's a knockdown of cancel 2 or knockdown of cancel 3 or a knockdown of Cat8, and we did gene expression analysis. And when we did that, we found about 173 genes, about 50% of them were actually repressed when we knocked out cancel 3 in these cells, and these were overlapping with cancel 2 as well. Of those, we found 27 of them, which actually been scored as being essential genes if you go to the DepMap database. So 27 of the 146 genes are actually known to be essential. If you look at what type of genes they are, mostly they're housekeeping genes. Some of them are actually enriched, so very strongly enriched for mitochondrial translation elongation. And what we found that when we reproduced or basically show that they are indeed essentially in our system as well, when here some of them are tested over here to the right, when you knock out some of the genes which we found here, they are indeed essential. So it seems like the NSL complex is required for the expression of a set of essential genes, which are essentially in human cells. So are the NSL complex target genes actually regulated by H4K16 acetylation? Well, to do that, we looked first at H4K16 acetylation. We collaborated actually with Epicypher to make sure that we had very specific antibodies. H4K16 acetylase is highly abundant modification shown enrichment over gene body regions, but it is actually highly abundant in the cell in general. We see that H4K16 acetylation, this is proteomics data, is associated with about 30% of all H4 in mammalian cells. And this is across a number of cell types. So when we looked at what happens to H4K16 when we knock down cancel 2 and 3, the picture actually changed quite a lot. So when we deplete Cat8, which is shown here, we completely lose K416 or H4K16 acetylation. When we deplete cancel 2 and cancel 3, we don't see any effects whatsoever on H4K16 acetylation, which is shown here in the bulk, but it's also shown when you look at transcription. And basically what we see is that this transcription is actually changing overall of the gene body, and we see perhaps a slight enrichment in the transcription start site. Sorry for that. So on the other hand, when we knock out MSL1 of the MSL complex, we completely lose H4K16 acetylation, which is shown here. So H4K16 acetylation in mammalian cells is actually catalyzed by the MSL complex together with Cat8. So the question is then, how does the NSL complex then regulate target gene expression if it's not through H4K16 acetylation? And one of the things which were interesting was there was a previous report published 10 years ago, which shown that the NSL complex also can catalyze H4K5 acetylation and H4K8 acetylation in vitro. It was never shown in vivo, and actually people who tried to never showed an effect of Cat8 on H4K5 acetylation or K8 acetylation in vivo. So what we did again was to characterize some antibodies to check their specificity on the epicycloarrays, and we found some antibodies which turned out to be relatively specific for H4K5 acetylation and H4K acetylation. And we actually mapped the sites, and they are sitting on transcription start sites. Furthermore, what we did was to look at what happens to H4K5 acetylation and H4K8 acetylation in cells where we knock down cancel-2, cancel-3, or Cat8. And what is shown here is that you basically see a strong reduction of those on transcription start sites on NSL target genes, which is shown over here as well. So very strong downregulation, but it seems to be some remaining in some of the genes. So how does it actually regulate transcription that complex? Well, to do that, we actually took use of a system where we very fast could degrade cancel-3. So in this way, we can use the primary effects of actually degrading this NSL complex on gene transcription, on proliferation, and so on and so forth. So here's an example of this. Here's cancel-3. We treat it with a DTAX for targeting for degradation. Within 2 hours, it's gone. When you look at long-term phenotypes, you actually kill these cells as well. So this is done in mouse embryonic stem cells. Furthermore, what we did was to look at some of the genes which we know from the other cells we used. They were actually regulated, but since they were conserved genes and also required for normal proliferation, they also seem to be regulated in the embryonic stem cells, which we verified. So now we are downregulating cancel-3, and we look at H4K5 acetylation and K8 acetylation. As you see here, it's taken down extremely rapidly. This is 2 hours after actually using the DTAG. So we basically degrade the complex, and you see a very fast turnover. You actually see a strong downregulation of K5 acetylation and K8 acetylation. So what we look at was happened with RNA Pol2. We see it's very similarly the same targets. We see it down, basically decrease in RNA Pol2 regulation, whereas we don't see that on housekeeping genes or other housekeeping genes. And again, what we see here is actually a reduction in expression. So this is looking at nation transcription. We see a very strong reduction in transcription of these genes very early on after the treatment with the DTAG. So it seems like that the NSL complexes catalyze K5 acetylation, K4 acetylation, and this way is actually promoting RNA Pol2 transcription and the early transcription initiation of these housekeeping genes in these cells. So what is the function then one could say about K5 acetylation, K16 acetylation in our cells? Well, so we went back and then we now looked at what happens in this situation. So when we knock out Cat8, we show over here, you lose almost, I mean, at least there's no detectable H4K16 acetylation left. We took 2 different guidanase to MSL1. We see a very strong downregulation as well of H4K16 acetylation. MSL2, despite being very efficient in knocking out MSL2, does not affect K16 acetylation in these cells. MSL3, we have one not very efficient oligo here, and then we have a very efficient guide RNA here. We can see this K16 acetylation is gone. What happens to embryonic stem cells and THP1 cells? These are THP1 cells. When you knock out MSL1, they don't care. They just grow. They go on. And it seems like that K16 acetylation is not required, at least in the mammalian cells we've tested so far. So going back to chromatin accessibility, here are a couple of our, let's say, negative results for that. So there's an micrococcal nuclease assays to look at bulk regulation of K16 acetylation for basically for the chromatin accessibility. We don't see any difference. When we look at ATAC-seq genome-wide, we don't see wide differences as well here either. What we then did was then actually look at where K16 acetylation is localized. So this is genome-wide. What you see up here actually is A and B domain. So looking at chromatin territories, A and B domain. And this is ATAC-seq, which is actually obviously localized with A domains. And what we see the same is actually with K16 acetylation. We see these localized, these mega-base domains of open chromatin as well. And we see a preferential binding in this area as well. So K16 acetylation is basically a very strong mark for this open chromatin, in particular for chromatin territories of the A compartment. So with that, I would like to conclude. This is a paper we published earlier this year in Molecular Cell, we call complex-dependent histone acetyltransferase activity of CAT8, MUF, MIST1, determines its role in transcription and cellular homeostasis. So what I've shown is that the NSL complex, but not the MSL complex, is essential for proliferation of human cells. This has been shown in Drosophila as well, and it's shown that it's actually but not required for the male germline, but only not for the female germline, but for the male germline for the MSL complex. The NSL complex catalyzes H4K5 acetylation and H4K8 acetylation at transcription start site, promoting transcriptional initiation. H4K16 acetylation is highly abundant modification placed by CAT8 and is part of the MSL complex. H4K16 acetylation marks open chromatin, but does not affect global chromatin accessibility. There might be redundancy here, we don't know, but at least H4K16 acetylation is not required. And H4K16 acetylation, interesting, was recently demonstrated to stimulate .1L H3K27 methylation is something we're looking into, a paper which was published in Science earlier this year. So one of the things which is still an open question is, what is the function of H4K16 acetylation in mammalian cells? And this is the model that we're basically defining the two complexes. The NSL complex has a role here, regulating transcription of transcription start site of housekeeping genes, where we have the MSL complex, mainly known for dosage compensation in Drosophila, but not in mammals, where it seems that it makes these big blocks of H4K16 acetylation, and we still have to find out what these are doing. So with that, I'd like to end my talk here by acknowledging the people in my lab who have done the work. The first part, in particular, Anditya Sankar, Faizan Mohammed, they were helped a lot by Hua Wang in my lab for the bioinformatics analysis. The second part of my talk by Alex Ratsitskoyskaya, who's done the major part of the work. She's been helped by Dasha Shlyarova for the bioinformatics, has also some help from others in the laboratory. In particular, we collaborated on that project by a very talented postdoc in Paul Chilea, who started in the old Nørgaard Jensen's laboratory, but actually moved to the MSK proteomics facility run by Ron Hendrickson, who's also been held on that project on the bioinformatics by Vasily Grinev, Richard Koch, and Yin Shang. And then finally, we collaborate on that project also with Epicipher for the validation of the antibodies. And with that, I'm ready to take questions. Thank you very much.
Thank you. That was wonderful, Kristian. We already have some questions coming in. So from Erna, she says, great talk, Kristian. Did you try making primordial germ cells after Epi-LC induction with your K27R mutant ESL to see if they perhaps form PCGs more easily than wild type?
No, we haven't. It's a very good question. It's not something we've actually worked a lot on. So our study here was mainly mechanistic, and we actually want to make a mouse to see what happens there. We've done that before because it's a very strong assay to actually use. If you do complementation assays and see early development, it's a bit stronger than embryo bodies. But I couldn't convince the people in my lab to actually do that. But we didn't do the primordial germ cells. It's a very interesting suggestion.
Okay, and Tamer asked, do CAT8 and MSL3 knockouts have different effects on cell viability? If so, what might be the mechanism?
Well, so is CAT8? Well, I'm not sure how to do the experiment. When we knock out CAT8, it's essential, whereas the MSL components are not. And the reason why CAT8 is essential is because it's also required for the NSL complex activity. So I'm not quite sure. Perhaps you can write in, how this experiment could be done.
Yeah, that would be tricky. One thing I was wondering is, have you looked at some of the other histone marks? And this pertains more to the first part of your talk, where in the absence of K27 trimethyl, does K9 ever come in to try and compensate? I know normally they never like to play together, but I was wondering, in this dramatic case, does the cell sort of adapt and use different repressive mechanisms?
It's actually something we're looking at in another project. We haven't seen big changes in H3K9 in those cells.
Okay.
But we are, so this is coming to, there's a project we're working on in the lab, which is kind of addressing, I can tell that, the histone code idea about, specificity. Can we bring in K9, for example, that Polycomb targets, and then see,
cool.
Can you actually now figure out you can rescue the phenotypes you're looking at? And it's been a little more tricky than we thought, but we think we have a model now in embryonic stem cells where we can actually put in K9 exactly at the K27 sites. It has some caveats because K9, as we know, also can be involved in setting up DNA methylation.
Right.
And the question is, how reversible is it? So we are primarily looking at ES cells, proliferating ES cells rather than differentiated ES cells.
Yeah.
Gene expression.
Cool. I look forward to hearing about that soon. And then we have another question here from Samir. Very interesting talk. Does CAT8 knockdown rescue differentiation of EZH knockdown in mouse organ progenitors?
Oh, I haven't done that experiment. That I don't know. I'm thinking here, and why would that work? I mean, we haven't, I can tell you one thing we've done, it's the result just went out of the lab, right? So we tried to rescue ring 1A, ring 1B knockout cells to see whether there would be anything rescuing that in embryonic stem cells by doing CRISPR screen. That didn't work. I can tell you that much. The only thing we got out of was a cereblon. We got cereblon because it was used to target one of the ring 1 components. The question is for the EZH2, because I think that's what you asked for. So EZH2 can actually proliferate EZH1, 2. They proliferate finely in defined media, no problem. We do see upregulation. So we don't need a rescue there. The question is whether it can rescue a differentiation, and we haven't done that experiment.
Great. Okay. And then since you finished early, we have more time for more questions. So Tamer is following up. Is there a CAT8 mutant that can interact with NSL but not MSL?
Well, I would like to make that one. That's a very good question again. Yeah, no, I don't know of it, but it would be an interesting one to make, because one of the things, of course, that I didn't mention very well, because another thing is that MSL and basically subunits, they regulate not only different activity but also different localization, and it seems like this is also part of the mechanism. Yeah.
Okay, and then Genevieve asks, what AB compartmentalization looks like upon CAT8 inactivation? Well,
very similar, but there you have to remember that the cells die, so we haven't done the experiment. The cells die when we knock out CAT8. So we cannot do it as clean as we can do with the MSL 1 or 2 knockouts. MS1 and 3, we did it with MSL 1, sorry for that. Yeah. Because they go fine, and we reduce K16 acetylation completely in a similar way as we knock out CAT8. But the problem is when we knock out CAT8, so CAT8 is an essential gene for these cells, whereas MSL 1, 2, and 3 are not.
Okay. All right. Well, thank you very much. And I have the awkward thing of saying I'm next, but I'll hand it over to Rick.
All right. So
thanks again, Kristian.
Thanks, you. Good to see you again, John. Take care.